All right, hello everyone. My name is Optine, and this is going to be a video on DNA and RNA structure. It's going to be my very first video, so hopefully it's not going to be that bad. But without further ado, let's get into it. Okay, so let's start off with DNA and its technicalities, specifics about it. So what is it composed of, and what is it? So DNA in its long name is called deoxyribonucleic acid. It's a pretty long name, but let's break it down. So deoxyribo, that comes from the deoxyribose sugar that is composed in DNA, that DNA contains. It is also a double-stranded helix, as you can see on the left side of my screen. I hope you can see my cursor there, but it has a 3' 5' end that is coiled around. And also you see those bases, right? G, C, T, A. Those are called purines and pyrimidines, and I'll get into the specifics of those two. What's the difference between purines and pyrimidines? But each purine and pyrimidine is made up of a nitrogen base, a phosphate group, as well as a pentose sugar. So let's dive deep into that aspect of the purines and pyrimidines. So if you see here in the middle, again, I hope you can see my cursor, you can see thymine, adenine, guanine, and cytosine. The yellow, let's examine thymine. Where it says thymine, basically, that's the nitrogen base. So thymine, adenine, guanine, and cytosine are all the nitrogen bases. Attached is this small ring structure, which is the sugar, the pentose sugar. And the reason why it's called pentose is because of the five side characteristic. And you can see that also there's a phosphate group, which is that central P with oxygens attached. And going back to the double helix diagram that I have there, if you see there are these dashed lines connecting, or that shows a connection between the GCTA and the bases essentially. Those are what are called hydrogen bonds. So the rungs of the ladder, as I like to call it, which are again the bases, GCTA, it connects the two strands of DNA because the nitrogen bases are connected by hydrogen bonds. They are very weak bonds, but it gets the job done at least. But now let's reverse back to the central diagram I have there, where it shows again the thymine in the yellow. If you see here the pentose sugar, where it is attached to thymine nitrogen base, it's attached by a bond as well. And but what is that? It's called a covalent bond. And again, the sugar and the phosphate are called, or connected, I should say, by covalent bonds as well. So covalent bonds are much more stronger than hydrogen bonds, but what you really need to make sure you know is that the nitrogen bases are connected by hydrogen bonds. And lastly, let's take a look at this table I have below my bullets. You see here I have purines and pyrimidines, purines on the left, and below that in parentheses it says double ring, and adenine, guanine are composed of that. Adenine and guanine are found both in DNA and RNA, as you can see. It said that in the parentheses, but I want us to examine the structure. You see here, it is this like double ring structure, right? And it looks like it has that, kind of like that pentose sugar characteristic to it too. But the thing is, if you compare it to pyrimidines, which is a single ring in parentheses, it is bigger because it's a double ring structure, right? And also the pyrimidines are composed of thymine, cytosine, and uracil. But make note of that only it is found in RNA. And I'll dive deep into RNA and kind of examine that. But in hindsight, just in general, you need to know that purines, adenine, guanine, and pyrimidines in DNA are going to be cytosine and thymine. Also, another important aspect about DNA is that it runs in an anti-parallel fashion or nature, basically. So for example, you see here that DNA in this strand, in this side, is going from a 5' to 3', and then from the right, it's going from 3' to 5'. So basically, DNA is not parallel to its other strand, to its complementary strand. But how do you know whether one side is a 5', one side is a 3'? If you see here on the left, there is a 5' end label where there's a phosphate group. If there's a phosphate group, a terminal phosphate group is what it's called. That's called a 5' end. That's how we can distinguish between whether one side is 5' or whether one side is 3'. And yeah, this antiparallel, as I mentioned here, they run in opposite directions. And just for example, if you have a 5' end with a phosphate group on the left, then you should know that on the right, on the other strand, it should be a 3'. Start with a 3' end, and then to 5' and vice versa. So again, just know that DNA is antiparallel. Now let's examine RNA. Very similar to DNA, but one big observation you can make is that it's only single-stranded. And also, as I mentioned 2 slides ago, thymine is not in RNA. Instead of thymine, we have uracil. But that is a pyrimidine, actually. So yeah, uracil replaces thymine. It is also called ribonucleic acid in its long-term name. And the ribo comes from the ribose sugar. So it has a different sugar than DNA. And as I said, single-stranded. And it has a similar sugar phosphate backbone. And we'll dive deep into the sugar phosphate aspect of RNA and both DNA because they both have it. But yeah, keep in mind 2 key aspects of RNA, single-stranded, and it has uracil. And now let's dive deep into the sugar phosphate aspect of both DNA and RNA. So on my left here, you can see the bases again, and those dashed lines, which represent the hydrogen bonds between the bases. And on the sides, we have the S labeled and a P labeled. Again, hope you can see my cursor. But that represents the sugar phosphate backbone because the S is labeled as sugar and the P is labeled as a phosphate group. So you can see on the sides of the DNA structure, it's all SP, SP, sugar phosphate, sugar phosphate. And that's what the sugar phosphate backbone is. It's just the sugar and the phosphate group of the nucleotides that are attached to the nitrogen bases. And for example, if you go back to, let's go back to RNA, this whole side here of RNA, excluding the rungs, the bases, right? That's all the sugar phosphates of those nucleotides, those nitrogen bases that are attached to sugar phosphates. And I have this diagram here that shows and conveys the central dogma of biology. We'll be covering that next. But yeah, just giving you a glimpse of what we'll be talking about, transcription and translation. Now, just for a recap of everything that we've talked about, I created this like really mini concept review thing. So first, a very important thing to consider is that DNA is double-stranded and runs antiparallel. The RNA contains all the bases except for thymine. It just replaces it with uracil. Purines are adenine and guanine, while the pyrimidines are cytosine and thymine. That is in DNA, and then thymine would be replaced by uracil in RNA. The sugar phosphate backbone makes up the sides of both RNA and DNA, as the rungs are the nitrogenous bases. And with that being said, thank you so much for watching my video. I hope it wasn't that bad, but please, I value your feedback. So in the comments, just let me know if I did well, if I did not so well. Let me know if I could improve on something, and I will definitely take note. And also keep in mind that, again, I'll be talking more about biology next in my upcoming videos, but I will also be making videos on physics, calculus, English, environmental science, and even SAT. I have some tips for that as well. But until then, thank you so much. Hit that like and subscribe button. That really helps as always. And I'll see you in the next one.